there was any direction of codon bias in our data set that correlated with expression. And the way we approach this is use a multivariate regression. We use a method called partial least squares, where basically the input is the individual codon frequencies and the expression data, and the output is an optimal set of weights that best predict expression. And what we find is that not only for each of the individual gene sets, but for both simultaneously, we can fit one regression set of weights that can predict expression for both sets. And the model that we get is highly predictive. If we leave out 20% of the data in the regression predicts that left out data with a high correlation coefficient. We also see that when we make genes that are predicted to be high expressing and low, indicated by the green triangles here, that the model accurately predicts their actual expression levels fairly well. So we're getting a handle on on at least what correlates with expression in these sets. The question is, what's the underlying biochemical basis for this? And we've got several experiments ongoing to ask this. If we look at the data, though, and compare what's going on with our best expressing genes, we do get some clues that maybe what's going on here. It turns out for our best expressing, or it turns out for the antibody gene set, that most of expression can be correlated to codon biases of serine and threonine. Elf et al. In 2003 proposed that certain tRNAs tRNA isoacceptors are more or less likely to become uncharged under conditions of amino acid starvation or accentuated protein synthesis. And what we find in our data set is that the best genes prefer codons that are decoded by tRNAs that are least sensitive to becoming uncharged under stressed conditions. So perhaps these tRNAs are more likely to remain highly charged under the stresses of heterologous overexpression. So we want to push the same type of study into other organisms beyond coli. We certainly want to add to our coli data, certainly with more proteins and more studies to get at the underlying mechanism, but we also want to look at as many different organisms of interest as possible. This slide represents a collaboration we have with Dr. Robert Stroud at UCSF, where we're looking at the expression of a human membrane protein in yeasts. And for this library that's shown here, we're biasing the codon bias for individual amino acids, either toward higher frequency or lower frequency, or toward higher frequency along the trajectory of the bias already in yeast. So we're including more of the high frequent codons in yeast, or we're suppressing those and increasing the level of infrequent codons. And we do this systematically across the variant set and look at what we get in expression. And like with the E. coli set, we see a wide range of expression levels. In this particular case, the protein, or the wild-type sequence gene, expressed undetectably. But our best expressors in this set give expression levels of up to a mg per liter in the membrane fraction, which is plenty of enough for them to take onto crystal structures. So they've gone from nothing to having a real chunk of stuff to work with. What we see also indicated in the red arrows is when we apply more standard optimization strategies, matching the bias of yeast or biasing toward high frequent codons across the board, we see very underperforming genes. So we have a method here of at least sampling some genes that are superior. And like the E. coli data, and this is still early work, but we can fit the data to a codon-based model using PLS that looks very good in cross-validation. We're adding more proteins to this set. We've currently got some data that we're crunching through now. We're pretty encouraged by our early results. So in conclusion, we think we have a good method, and I think the method is represented by both ours and Dr. Plotkin's work, a really good way of sampling essential variables related to expression. And in our data set, in contrast to what others have seen, we do see a correlation with codon usage, at least something that we can tie to codon usage. The underlying basis, I think, is still a mystery. But maybe we have some clues, and maybe it's related to tRNA sensitivity to overconsumption. We want to push this into as many organisms as possible. We want to see is what we learned general. We also want to expand to as many different proteins as possible to see if it's general across different protein types. We have ongoing studies in many organisms right now, and hopefully we can get some more of that out into the literature soon. And I'll stop there.
Great. Thank you very much, Dr. Welch. So thank you all again for the excellent presentations, and we're going to move right on to the Q&A portion of the webinar. We've received a lot of questions, so we're going to move quickly to that. Just a reminder to everyone that you can still submit your questions by simply typing them into the Ask a Question box and clicking the Submit button. So the first question I'm going to pose to you is it better to reduce the probability of mRNA secondary structure on the whole mRNA, or could one just get away with avoiding secondary structures in the first 50 to 100 nucleotides and still get a similar translational speed? So, Dr. Plotkin, should we start with you?
Well, thanks very much, Sean. Interesting question. I think we certainly have some data that speaks to that, but I bet all of the other panelists will also have data that speak to the same kind of question. In our study, it was pretty unambiguous that it's the structure in the first 50 or so nucleotides that is most important. We certainly had a lot of constructs amongst our 150 that had significant structure on the inside part of the gene, but nonetheless, even when there was structure there, they were expressed at levels that were dominated by the 5' end. So, at least in our data, the structure of 5' is most important, and that's consistent with reports that the ribosome can progress fairly easily through large structural elements, whereas initiation is much more structure dependent.
Yeah, so we can confirm that for the human data. We haven't looked at the structural features in as much detail as my colleagues here, but we examined the presence of secondary structures in the 5' UTR and the 3' UTR and in the coding strand. And the 5' UTR, the secondary structures that was the only place where it did matter, and it influenced the protein expression levels. And also, those were the nucleotides closer to the translation initiation site than further away from it.
We also looked at internal structure in our data set. And in fact, we've made genes among the antibody and the polymerase gene sets with intentionally very strong internal structure to see if we see an effect. And we've never seen any effect of doing that. Now, I wouldn't go so far as to say that any structure, that no structures would have an effect on elongation of translation. And I think certain types of structures, in particular strong ribosomal or strong RNA pseudoknots, could induce frame shifts and maybe create pauses, and that may be accentuated by sequence context as well. So I wouldn't go so far as to say you should remove all structure in a sequence. I think that's restricting yourself too much in design. But I think it's more nuanced. It still may be involved.
Great. So our next question asks whether optimizing a gene sequence can affect folding of the mRNA and the encoded protein, or the encoded protein. So anybody like to tackle that one? Dr. Plotkin?
Well, so I'm not so sure we have data that speak to the encoded protein itself. We certainly observed that the choice of synonymous mutations can affect the folding of the mRNA. And I guess there are anecdotal reports that as a function of the mRNA sequence, even synonymous sites, there can be ribosomal pausing that can also affect the folding dynamics, if not the eventual fold of the protein. But that's not something that our data particularly speak to. I don't know if you guys have.
I wouldn't be able to add anything. The amino acid sequence plays a role, but if that doesn't affect it, I wouldn't be able to think about how the stability of the protein is affected.
Mm-hmm. There are a number of anecdotal reports that I think pretty clearly show that coding sequence can affect how a protein folds. In one case, a recent report where the enzyme specificity is influenced by synonymous codon changes. So there could be definitely a role there, and I think it's just a mystery or it's a question of how we approach it systematically to study it. But I think that definitely that's a good place for future study.
So this is a question I think for Dr. Vogel. Do you have any comparisons of protein stability between older proteins in evolutionary terms and newer sequences?
Protein stability. Well, there are very few data sets, large-scale data sets on protein stability out there. There's one large-scale data set from the O'Shea lab on yeast, and then there's a recent one for human cell system, but that has some technical difficulties to it. So it's a hard question to answer on a global scale. So evolutionarily conserved proteins tend to be more highly expressed. We don't know about the causality between those two, so whether they're conserved because they're highly expressed. So then they also tend to be more stable. So that is kind of the generic answer to this. The details of that question depend on the system you're looking at.
Okay. Anyone else want to add anything? A complete answer for us. Great. So here's a philosophical question for you. Why does codon bias exist at all? Do you have any theories on in evolutionary terms, why it would have come about? We'll start with Dr. Welch.
Sure. I think there are a number of ways you can imagine the evolution of codon bias, and many of these have been analyzed. And I'm certainly not an expert in this field, but pursue the literature, and you'll find a lot of discussion on this. Biases in general GC content would affect codon bias, and there may be evolutionary purposes for that. Biases that affect your mutational direction I think are interesting to propose. So certain codons are more likely to yield different amino acid substitutions than others when randomly mutated, and that may be evolved. There's there are what else? So we've if we have any...
Well, so, yeah, I think maybe another approach to the exact same question is, there's this classic theory that the relationship between the codon bias and the expression level, and this is something we've discussed in our presentations a few times, but that is worth mentioning again, that led to the belief, among some at least, that a high codon bias matching the tRNA pools, which may have drifted to particular abundances on their own, will causally produce high expression. And we were both surprised to find that did not happen in our data sets, and in retrospect, maybe we should not have been surprised. The idea that codon bias exists as a mechanism to promote high expression is a bit far-fetched compared to just the possibility that a few sites in the promoter can be more easily changed to affect expression. So the particular abundances of the tRNAs may have drifted randomly, and I would suggest that the codon biases observed in the most highly expressed genes have evolved in response to their exogenous expression level more than as a mechanism to produce their expression level. So at least we have some feeling for what is not a reason why codon biases exist. Mm-hmm. Exactly why they exist, I think, is still open. Mm.
Yeah, and people have seen different biases among different classes of genes, and the proposal is that genes that are turned on under certain environmental conditions are biased according to what the stresses of those conditions might be. So you'll see some of this in the ELF paper that I cite in an earlier slide as well. They propose, they've seen that also tRNAs or genes that encode for amino acid metabolic genes, synthesis genes, are biased toward codons that are read by tRNAs that are less susceptible to amino acid starvation. So they propose that you have bias that basically corresponds to the environmental conditions that you're dealing with, and maybe bias has evolved as a way of giving you maybe even a sensing mechanism. You have a bias that's maybe extreme, and you can sense by way maybe of your tRNAs becoming uncharged that things are wrong, and then you can trigger some other genes that have a bias more appropriate to that condition. So I think there's a long way you can go with this. I agree with Joshua that it seems backwards to expect that bias has evolved as a control mechanism for protein expression. You don't want to control how much protein you're making by stalling your ribosomes on the message. You ideally would want to control by how much message you make and how fast your proteins degrade, as Christine was talking about. Why control the intermediate steps?
It sounds like a topic we could have a webinar on just by itself. So I'm going to come back to stability, question of stability, but this time about RNA. And the question is, what role does RNA stability play in expression levels, and are there any methods to control for this? So, Dr. Vogel?
Yeah, I can start on this, but I'm sure I'm not the only one who has an answer. So, in our human data set, we also looked at experimental data sets from other studies where mRNA stability was assessed. And we did see an inverse correlation between the amount of protein produced per mRNA and the mRNA stability. So that is important to distinguish because we look at steady state levels. So the mRNA stability itself should not influence the number of proteins per mRNA, but there are co-regulated. And the second thing we observed, we looked at the prediction of polyadenylation sites, and again, there was a correlation in our data set that speaks for an influence of mRNA stability on protein levels.